Hello, today we're going to talk about how muscles help us move and how neurons actually innervate with those muscles. Which of the following offers the best description of neural transmission across a mammalian synaptic cleft? The answer is C. Neural impulses cause the release of chemicals that diffuse across the gap. Muscles is what we're going to talk about today. Muscles always move by contracting. They're always shortening. And they have to be attached, of course, to some sort of skeleton, either an endoskeleton, exoskeleton, or hydrostatic skeleton. Muscles are usually antagonistic. So in order to move one way, one muscle contracts. In order to move the other way, another muscle contracts. So for instance, in your arm, biceps contract to move up your forearm, and triceps relax. And in order to move your forearm down, the biceps relax and the triceps contract. If you look at the grasshopper leg as well, it has these extensor muscles that are relaxed when the flexor muscles contract to move the tibia close to really the femur. To extend the tibia, the extensor muscles contract and the flexor muscles relax. Muscle cells are made up of myofibrils. The myofibrils are composed of two kinds of filaments. Thin filaments, which are composed of actin and a regulatory protein that wraps around the actin, known as tropomyosin, and thick filaments. Thick filaments have staggered arrays of myosin molecules. So skeletal muscle is made up of multiple muscle fibers. And a muscle fiber is actually a single cell that's fused during embryonic development. The myofibrils have contracting units referred to as sarcomeres. The sarcomeres are basically, again, thin and thick filaments. And these thin and thick filaments slide past each other. The thin and thick filaments, there are areas labeled, which I'm not that concerned that you know, but the A band is where all of the myosin is. The I band, you'll see that changes really in size. That's where we just see the actin here and no myosin covering. And the H zone is where we just see the myosin, the thick filament with no thin filament. So the sliding filament model of muscle contraction is the sarcomere actually shortens in size. And the way it shortens in size is thin filaments actually slide to the center. As a result, the I band and the H zone shrink. The way that they slide is the myosin has these myosin heads that interact with the thin filament. When ATP binds to the myosin head, it causes the myosin head to be in high energy configuration. That allows it to make a cross bridge with the thin filament, these binding sites on the actin, known as just myosin binding sites. When the ADP and the phosphate fall off of the myosin, then it changes to a low energy configuration that pushes the actin or the thin filament towards the center of the sarcomere. You can see that over here as the myosin heads grab the thin filament and push it along. Well, in order for muscles to control whether or not this sliding filament model functions, they need a way to turn off this sliding mechanism or turn it on. So the way that this works is the thin filament is actually surrounded by a regulatory molecule known as tropomyosin. Tropomyosin covers up those myosin binding sites normally when there's no calcium around. On the tropomyosin, there is this troponin complex with calcium binding sites. When calcium is available, it moves the regulatory protein, tropomyosin, out of the way and opens up these actin binding sites so that myosin can bind to them and can cause the sliding of the filaments and cause the sarcomere to contract. Which of the following does not form part of the thin filaments of a muscle cell? It should be D, myosin. Okay, now in order to control whether or not our skeletal muscle contracts, we have neurons that innervate or interact with the muscle. So this is a diagram of one muscle cell with a motor neuron axon terminus interacting with it. This is the synapse really, and there's a synaptic cleft between the two. Inside this cell, there is a huge specialized endoplasmic reticulum known as the sarcoplasmic reticulum that actually stores a lot of calcium that is released in order to allow the muscle to contract. At the end of the axon terminus, the action potential runs down to the end of this axon terminus. It causes voltage-gated calcium ion channels to open. Calcium rushes in, causes then the exocytosis of acetylcholine into the synaptic cleft. So acetylcholine then binds to ligand-gated ion channels. Those ligand-gated ion channels cause an action potential to be passed along the muscles to these T tubules or transverse tubules. That action potential then is sent along next to the sarcoplasmic reticulum, and that causes voltage-gated ion channels to release calcium into the cytosol. When calcium is released into the cytosol, it then binds to the tropomyosin, to the troponin complex really of the tropomyosin, causes the tropomyosin to move out of the way, exposing the myosin binding sites on the thin filament. The myosin head then can bind when ATP is hydrolyzed, and it can push along the thin filaments and cause contraction of the skeletal muscle. So during the contraction of a vertebrate skeletal muscle fiber, calcium ions. The answer is B, bind with troponin, changing its shape so that the myosin binding sites on actin are exposed. So in order to control the muscle and have fine motor skills, fine motor movement, the whole muscle actually is graded. There are graded contractions. Now, the graded contractions depend on the number of fibers that contract and the rate at which fibers are stimulated. So like if you want to pick up your pencil versus lift up a desk, lifting up a desk requires more number of fibers really that contract and require a higher rate at which the fibers are stimulated. A muscle fiber is innervated by one motor neuron, and each motor neuron may synapse with multiple muscle fibers. So you can have one motor neuron interacting with actually multiple muscle fibers. If you want to have a weak contraction, you could have a few of those muscle fibers turned on by one motor neuron, or you can have two motor neurons actually interacting with multiple muscle fibers, and so you can get this whole unit here contracting. So this graph actually shows how multiple action potentials sent by a neuron can cause summation or graded contractions in our muscles. So you can see if there is one action potential sent along a neuron to a muscle, it can cause a single twitch. If two action potentials are sent close in time, it can summate and cause a stronger contraction. If you have multiple action potentials sent by a neuron frequently within a short amount of time, you get the sustained contraction or tetanus, not to be confused with the disorder caused by the exotoxin released by bacteria. So which of the following is the correct sequence that occurs during excitation and contraction of a muscle cell? So the answer should be E. So there are different types of muscle fibers, different types of skeletal muscle fibers, and a lot of times we classify them as either slow oxidative, or fast glycolytic. Slow oxidative and fast oxidative, those are both using the electron transport chain, and they're using aerobic cellular respiration. Fast glycolytic is using just glycolysis mainly in those fibers. Generally in oxidative muscle fibers, we see that they have more mitochondria, that they have more myoglobin to pick up oxygen, and also they're darker in color. They're a red to a pink coloration. People talk about the meat in chicken being dark meat or white meat. They're actually referring to the different type of muscle fibers. The dark meat is actually the oxidative muscle fibers, and the white meat is actually the glycolytic that lacks a lot of myoglobin. Other differences include actually the number of capillaries and oxidative. In order to get more oxygen, they need more capillaries to bring that oxygen to them. There are some other differences that you can see in the table as well. So which type of muscle fiber would have calcium in its cytosol for a longer time? Slow oxidative, or fast glycolytic? Well, the answer should be slow oxidative. Slow oxidative could have calcium in its cytosol for a longer time because in order for you to have that aerobic muscle contractions, calcium would allow you to have more contractions for a longer sustained time, which means you require more ATP from cellular respiration. Cardiac muscle is similar to skeletal muscle in the way it contracts, except that there are intercalated discs, which are basically gap junctions that allow for a signal sent really from the AV and SA node from one muscle fiber to the next muscle fiber. So cardiac muscle passes along action potentials and actually generates those action potentials from the SA and AV nodes. Smooth muscle, remember, we find around a lot of organs, and it's referred to as smooth because it doesn't have a regular pattern of myosin and actin. The contractions actually can be initiated by the muscles themselves and don't need to be innervated by neurons, or they can be stimulated by the autonomic nervous system. Movement is found in a lot of animals, not all animals, and there really is differences in the amount of energy that's required to move. In swimming, organisms have to overcome friction, so a lot of times they have streamlined body shapes. And organisms that are on land, whether terrestrial walking animals or flying animals, have to support themselves against gravity. The energy cost of locomotion really depends on the mode of locomotion. And by this diagram, you can see generally with the body sizes that the energy cost of running is the most expensive at the same mass compared to swimming. So why are the muscles of an animal that has recently died likely to be stiff with rigor mortis? Well, rigor mortis is a sustained contraction. It's where the muscles stay contracted and can't relax. Now that's caused by a couple things. Number one, calcium remains actually in the cytosol of the skeletal muscle. Calcium, in order to be stored in the sarcoplasmic reticulum, requires ATP for active transport. And there's no ATP to pump it back into the sarcoplasmic reticulum. Also, it requires ATP for the myosin heads to actually release from the thin filaments. So why does rigor mortis actually end? Well, after the organism dies, lysosomes actually release enzymes that digest the myosin head and cause actually the breakdown of the interaction between the myosin head and the actin. Well, that's all for today, guys. Next up, I'm going to discuss more about how our senses function.